and it's a spinoff from some other community engaging work that we did in the past through a Center for Health Equity, through a collaborative grant with Moffitt and our College of Public Health. But we have our primary community partner here with us today in the person of Dr. Deborah Austin and Mr. Michael Morgan. And we're just so glad to have them. They assist us in reaching out and communicating with your group and others. And then Mr. Hiram Green, who's also affiliated with the university, but he serves as our community relations liaison as a part of our core team. And so this is but one of many of our pleasures as we come to you today with the opportunity to talk more about the pandemic and particularly the vaccine. Dr. Sneed and myself are pharmacists. He is an ambulatory care specialist, and I have specialized in things related to geriatrics and neurology. And outside of our care as pharmacists, we're both of us remain very passionate about things like the pandemic. But I do want to, as I introduce him to you, let you know that Dr. Sneed is one of a few pharmacists who not only has a pharmacy degree and has done postgraduate training to become an ambulatory care specialist, but he also went to University of Central Florida and has a degree in biology with a minor in microbiology. So he comes today truly trained and having a lot of insight about things related to the virus that most pharmacists don't. But then he and I have also been fortunate to work alongside all of the initiatives at the university that have allowed us to have both the COVID testing and COVID vaccine clinic. And so we're really excited because it's allowed us to work with the community in this fashion. This is probably close to about 40, if not more, of a series of workshops, town halls, podcasts, interviews, panel discussions, and other mechanisms that we've done for the last several years to talk to the community, particularly Black and Brown communities, about things related to research and opportunities for them to appreciate, a lot of the things that are happening with those processes. But we also pride ourselves in educating the community about disorders that impact diverse groups and help them understand, again, what's going on. And this pandemic has given us a lot of opportunity as we've gone from understanding the disease to embracing what having vaccines would look like, and then now to be able to talk about what we're seeing now that we've started vaccinating individuals. So today, Dr. Sneed is going to give you a very short presentation, and we have received quite a number of questions from others prior to today's meeting. And so what we're going to do is try to walk through those questions, and we'll encourage people, if you would utilize the chat. Dr. Austin and I, with the help of Ms. Jackson, will try to make sure that we man the chat and give appropriate attention to the questions. And so without further ado, I would like to introduce my boss, my dean, colleague, and friend, Dr. Sneed, and he'll tell you more about him as we kind of go further. So thank you so much.
Dr. Hill, thank you very much. I don't think there's much more to share. I think you very generously, and probably too generously, shared a lot of my background with our group here. First of all, I just really want to thank Ms. Jackson extensively for, just for her partnership and her willingness to hear from us today. And I can't tell you how excited I am just to even be working with this company with your name. Part of our foundation here, one of our pillars in our program is pharmacogenomics. And so just to work with a company that has the name genomics in it is really exciting for me. But today we're really going to talk about the coronavirus. I'm going to have a little bit of a primer in the very beginning to help people understand a little bit more intimately of everything that's going on with it. We're going to try and put it in lay terms for many of you, and then for some of the other scientists, you will probably want a little bit more, but we'll try and manage all of that together. So I'm going to share my screen here, and let me make sure it gets queued up appropriately and properly. And again, we'll just start off with a very short and brief presentation, and then we'll jump directly into a Q&A with all of you. Okay, so really just to get started on all of the hoopla that really has occurred over the past several weeks and months now has really been over the fact that, even though we had about 6 lead companies out there that were working on many of the vaccines, obviously 2 and soon to be 3 have really jumped out in the lead. Here we show that we had Moderna and BioNTech and Pfizer with their messenger RNA vaccine technology. Now, I'll share a little bit more in just a moment, but it's really important to understand that this technology is not brand new. That's one of the misunderstandings that many people have. It's the first time we've had a chance to rapidly deploy it, but we've been working on this technology. And when I say we, I mean all of us in the scientific community really want to give much more due to the people up at NIH and other groups that have been working on it in the laboratories for a long time. But starting back in, even as far back as 2013, so the technology platform is not brand new. Now, we didn't wake up in April of 2020 and all of a sudden walk into the kitchen and start trying to cook up a brand new vaccine. And that in reality, the messenger RNA technology, messenger RNA was discovered several decades ago, really advanced pretty well with the understanding of oncology, therapeutics. And then we had the bright idea to be able to take that and translate that over to vaccine therapeutics. And companies have been working on that for about an 8-year period. Now, very shortly, we are anticipating that Johnson & Johnson, or the Janssen company is appropriately named, they will be submitting for an emergency use authorization, or EUA, any day now. I'm eagerly waiting to get that data, because when they do submit, they will submit up to a 53 to 55 page report, the way the other 2 companies did, and I will extensively go through all of that data and get a better understanding of exactly how they did their clinical trials, where they did them, and some of the safety and efficacy data that comes out of that. And the last thing I'll say about either one is the fact that even though messenger RNA technology has been in the news very much, the Johnson & Johnson product will be what we call an adenovector, or adenovirus delivery mechanism, but really much trying to accomplish the very same thing in terms of encouraging the body to respond to spike proteins that will be messengered into our cells through the vehicle of adenoviruses. The next company after that, more than likely, will be Novavax, and they will actually be a protein-based vaccine, a little bit more traditional in a sense, but again, very new and novel technology. So here, again, I'd like to show this to the lay public. And the reason I love to show it, this is an actual photograph of a cell that has been infected by the coronavirus, by what we call COVID-19, the novel coronavirus. Now, this is a single cell in your body, and when we begin talking about replication and multiplication of these cells in your bodies and making millions of copies, this gives an example of what we're talking about. So the little yellow dots are the actual coronavirus on that cell. Now, many people across the country, through one means or another, whether it be social media or other misinformation, some people don't even believe it really exists. Well, yes, it does exist. We have an electron microscopy photograph here to prove that. And this photograph came directly out of the labs from Dr. Fauci and his group at NIH. Now, one of the most important things, and all of you have seen the photograph of the red spikes that are on the outside of the coronavirus. It's really important to understand that that became a therapeutic target for what we're trying to accomplish in the way of a vaccine. But the other part that most people have not really heard about is how important they are for infecting the body. All throughout our body, we have a receptor known as the H2 receptor. Now, all human beings have it. So this is a human virus. It's not isolated to one particular group or one particular race. And even children have the H2 virus, but it's not very mature and not as mature in children as we have it in adults. But the most important thing is that red spike that we all talk about, that becomes the attachment point and the entry point for the virus to attach and attack our cells, gain entry into the cell, and then replicate from there. And so I'm going to show you a video here in just a moment that will kind of explain that a little bit more. But many of the therapeutics that have been out there so far have really been focused on trying to prevent the attachment of the red spike protein onto the H2 receptor. And we should also really begin to understand that if you think about Velcro and like wool and how sticky that would be, this is exactly the type of attachment point that even the mutations that we've heard so much about have learned much more in how to more efficiently attach onto that cell, gaining quicker entry and then replicating at a higher rate, leading to higher transmissibility. Now that ACE2 receptor, again, is all throughout our body. Now back in January of last year, we all began to think it was another respiratory virus. And of course, it has respiratory in the name and SARS-CoV-2. However, that ACE2 receptor is in many organs all throughout our body. And it really helped us understand early on exactly how and or why we were having so many varied types of reactions and symptoms that were developing from people who were infected with COVID. And it wasn't just a respiratory virus. Very early on, probably in February of last year, we began to realize that people were having very significant diarrhea symptoms. Many of the people that were being hospitalized, that was one of the initial presentations. Some people that were admitted, that was the only presentation they had. They didn't even have a cough. Interestingly enough, unlike many other respiratory viruses that we're very commonly infected with, sneezing is not one of the common symptoms that we find with this particular virus. Here, you can see here, we have ACE2 receptors in the heart, we have them in the intestinal tract, we have them on the kidneys. And even though we have, they're not expressed in a great deal in the lung, because we have such a large surface area, that explains exactly why we have so many people with respiratory challenges, eventually leading to ICU, being placed on ventilators and having other problems. And so here, the thing I'd like to show here is the fact that we have a lot of different areas of the body that become infected. Number one, you've heard of people with the neurological problems, the loss of taste and smell. That becomes a really big problem and challenge. We found acute kidney injury early on. Down in the lower corner, I've talked about the gastrointestinal challenges. And also, but really began to emerge and let us know we were dealing with something maybe a little bit different, was the clotting that began to form. And we had reports that came out of Philadelphia in early March of last year, really talking about not only just clotting, but even the microclotting, clotting under the skin, clotting in diffuse areas throughout the body, where normally you wouldn't expect that to be occurring with many of the respiratory viruses. And then the very last component was really attaching onto the heart tissue and leading to conditions like myocarditis and cardiac arrhythmias. Again, very dangerous virus to really deal with. So one of the last two slides I have here, I put this in just for you all. I have a quick video I'd like to share with you.
In a world where millions of lives are under threat due to COVID-19, it is of vital importance to gain a better understanding of how the virus actually works in search of a cure. The surface of the virus particle is covered with approximately 100 spike proteins. They always come in groups of 3, in which the proteins are intertwined. Each individual protein consists of 2 parts: a globular head called S1 and a stalk-like structure.
Oh, I do apologize. I've been having challenges with that particular video lately, and for some reason it just doesn't want to stay on for me like it used to. I apologize for that. So we're going to try this one more time. And I think in short order, we'll just move beyond that one pretty quickly, just for time's sake, because I really do want to get to a question and answer with you all. But I can explain the remainder of what that video was sharing. So the focus of the vaccines, the focus of the vaccines really is to help your body build antibodies against those spike proteins. And here we begin to show that exactly what the antibodies, when they attach onto the spike protein, it prevents entry of that particular virus into a cell. But we need a lot of the antibodies to be able to attack so many of the various virus particles that I showed you earlier. And so the very last thing I'll share with you all, many people like to know and want to know, if I get the vaccine, what can I expect? Well, the first thing I would tell all of you, number one, the currently available vaccines, and this will not hold up for the other vaccines that come out, but the two on the market right now, both are preservative free. Okay, the messenger RNA vaccines have no preservatives in them. Now, I do not expect any of the preservatives that we have in any of the vaccines that come out to be dangerous or problematic for anybody. But because we don't have preservatives and messenger RNA is extremely fragile, so we have to keep them at very cold temperature. That's why with the Pfizer product, we have it at -80. And with the Moderna product, we have to keep it at -20 or normal freezer temperature. You can see here, you need a 2-shot vaccine strategy for both. And the overwhelming adverse effect or side effect that most people have really is fatigue. Just like as if you were being infected by a virus, but make sure we understand that when you get the vaccine, there is no virus that you can get infected with. You cannot become infected with the currently available vaccines. There are no live viruses in any of them that are coming out. You may experience headache. Some people have nausea, vomiting, although very small amount, chills, and then fever. Now, it's also important to understand, you see, it's really enhanced with the second vaccination shot and not so much the first. That's why I've highlighted both of them with red blocks here. And then the very last thing I will share with you, even though, if you see the percentages here, it's not 100% because I've talked to people that have no symptoms whatsoever. And it can easily be remedied by taking Tylenol if you begin to experience them. But in many cases, I've told my patients, just take the Tylenol ahead of time and don't even think about trying to let it come on even before it to be problematic. And so there, I'm just going to say that I personally, I've had the vaccine. I've had both shots. I have very minimal side effects. And then moving forward, I'm hoping that many more people will elect to take the vaccine. So I'm going to stop right there and just listen to your questions and really hopefully have a robust conversation with many of you. So Ms. Jackson and Dr. Hill, I'm going to let you all drive from here.
Very good. Well, thank you so much, Dr. Sneed, for that insight in regards to the vaccines. And I would like to invite everyone to put questions in the chat area. And we'll also go back and look at some of the questions that were submitted prior to, and we'll get started on those. So please feel free to put questions in the chat area for the dean to answer. So I'll kind of go back and forth. One of the questions that we got previously was, Dr. Sneed, how long does the vaccine provide protection?
Okay, well, at this current time, again, we're just approaching, we just passed the 1-year mark from really understanding that we even had COVID-19 to worry about. The clinical trials in phase 1 began in probably around late March, early April of last year. And well, what I can tell you is that the people that were the earliest to go through clinical trials still have they currently have antibodies that are being detected as being neutralizing. And so right now we're going to be pushing up on a year. We're expecting at least a year. There may be evidence that they may last as long as 2 years. But we won't know that for a period of time. And so the next question that most people ask after that would be, well, do we think it will become an annual vaccination shot like we get with the flu? And that's quite possible, especially if we have to begin to try and match up the vaccine to any of the mutated strains that may be coming out eventually sometime in the future. So it's very possible that we will have an annual strategy, not because your antibodies no longer work, but because we're trying to pair up to a new mutated version, which is the very same thing that happens with the flu virus right now.
Okay, very good. Ms. Jackson, would you like to take the first question in the chat?
Sure. The first question is, has the vaccine in trials been tested on autoimmune patients?
Yeah, I get that question a lot. That's a very important question to ask. First of all, when we talk about autoimmune disorders, there's a whole spectrum, an array of conditions that fall into that. And in terms of inclusion criteria, they did not specifically target or allow people into the clinical trials that had these particular conditions. Having said that, we've had a number of people around the world that have now received the vaccine that had autoimmune disorders, and many of the endocrine and rheumatological societies are now actually recommending that people try and get the vaccine. Part of the problem and challenge is the fact that it's not so much the autoimmune condition, but very often the medications or other treatments that may be used to treat those conditions is really giving us a cause to have to pause and think whether or not we want an individual that has an autoimmune disorder to get vaccinated, not because it will become possibly dangerous, but there's a great potential that you may not be able to mount the robust immune response to it. Having said that, many people around the world and even here in our country are beginning that we should go ahead and vaccinate people with autoimmune disorders, because you could become infected with the actual coronavirus. And if you were to become infected with the actual coronavirus, your body would make every attempt it could to try and mount a response to that particular virus if you became infected. And so many people are now beginning to think that we possibly want to take that chance, but we're pushing everybody back to their provider to really get their insight, because ultimately the individual treating that condition would have to make a decision about how they wanted to monitor you as an individual patient according to the individual autoimmune disorder that you have.
And just to add a little bit on that, I have a neighbor who has lupus, and she was saying that her doctors highly recommended that she go ahead and get it, and so she was ready as well. So a next question we had was, with us now probably getting close to having 3 or at least 4 vaccines by the end of the year, do you have one you prefer to suggest? And how do you compare the 2-dose vaccines to J&J's 1-dose vaccine?
Well, let me just share this. And again, and Dr. Hill, she kind of shared a little bit of my background. Before I got my doctorate of pharmacy degree, I was on my way to go get a PhD in immunology. So I really respect that the cancer work that you all do from a diagnostic standpoint. And it's really difficult to answer that question. Number one, I don't want to get into the business of trying to pick a winner or a loser in terms of which one I think is better or not better. I think it would be very difficult for one particular vaccine to be able to get to everybody. We expect to want to vaccinate about 270 million people in our country. Or, if you really think about it, we're probably talking about somewhere between 3 to 5 billion people around the world. Having said that, I think people should get, if we approve a particular vaccine, I think we should take what is available. I think the messenger RNA technology, I'm really excited about that. I think the technology was built to be deployed rapidly in case we had a pandemic. In the longer presentation, I'll go into the whole entire history of that. And but I also want to share with you the adenovector viruses that are coming out. The adenovector vaccines that are coming out. The technology is not that much different. I mean, we're trying to match up part of the RNA from the genetic code of that particular portion of the virus. In this case, the genetic code that matches up to the spike protein, and we're trying to find good and novel ways to quickly identify that and then introduce that into your body. I really want to share with you, it may be a little bit misleading if you look at the news, and that's why the reports and the peer review reports coming out are so important. Both the Moderna and the Pfizer vaccines, when they were going through clinical trials, did not have to encounter some of the new variants that are now emerging around the world. They only had to encounter the original wild type, as far as we know. And so if we're looking at lower rates, we also have to try and pay attention to how many people in that particular clinical trial for Johnson & Johnson, for example, how many of them came into contact with the new variant. And when we were using the genetic code from the wild type, the original virus, in terms of one that had not mutated. And so my recommendation right now is that you should get the one that's available when it comes out. Many people don't want a 2-shot strategy. For people that don't want that, I think the Johnson & Johnson will be a very adequate vaccine for you. And also, just to let you know, Johnson & Johnson has a second clinical trial ongoing right now that is a two-shot strategy with an initial one and then a booster to come right behind that. So I'm not going to say which one I would pick over one over the other. I think the messenger RNA technology is very exciting. We can come back and try and get match it up to emerging variants that may come out, and they're already doing that right now. But the ones that are out, they're very good. Also, the Novavax coming out, and in full disclosure, I am a clinical investigator for Novavax right now. I want to be very clear about that, but so far, they're reporting close to a 90% efficacy rate for their clinical trials thus far.
Well, looks like we've got several questions about Moderna. Ms. Jackson has one, and I think I have two. So I'll let you go ahead.
Okay. It says, I know someone who had a severe reaction to the first dose of Moderna. In this case, would you still recommend this person get the second vaccine dose?
In that case, I think it would depend on exactly what type of reaction they had. So far, and I haven't seen the Moderna data in the way I've seen the Pfizer data, but so far, around 21 people around the country have had very serious anaphylactic reactions to the Pfizer vaccine. I don't have the exact number. It may be similar with the Moderna product. However, it depends on what severe reaction they had. And none of the people, number one, have died. Most of the people recovered without too much of a problem. But 17 of the 21 had known allergic potential long before they took the vaccine. Many of them already had an EpiPen in their possession and still chose to go out and get the vaccine. And we have to kind of respect that. They want to be protected. And they trusted they would have supportive measures. I think about half of them were hospitalized. And so it would depend on what reaction they had. Now, for one reason or another, and I don't know if it's social media, Moderna has been getting a little bit more of a bad rap than the Pfizer product right now, but I can't confirm whether or not it's really true, but I am paying attention, and we'll be combing through FDA data here very shortly to figure out if we have a higher incidence of one versus the other. I can also tell you, and I showed on the chart, that a few more people did have more of a reaction in terms of the fatigue, in terms of headache and other conditions from the second Moderna shot, a little bit more than the Pfizer product.
So some additional questions were, with Moderna saying you have to get 2 doses for those people that may in fact have a severe allergic reaction or for whatever reason just don't get the second shot, is that a good thing? And is it the second shot that seems to be causing, most of the reactions we're seeing? And then the last piece is that there's information out there suggesting people who get the Moderna shot may actually need a third shot.
Okay, well, I'll come back to the third question. You hit me with three rapid fire, Dr. Hill. I'm going to try and remember them all. Let me address one quick thing right now. When people have the adverse reactions, okay, the common ones that we talked about, that is not a bad thing, okay? It makes us feel uncomfortable. We don't want to go through it, but it really is a sign that your body is now building the antibodies, okay? You're having, your body is now pulling energy in different areas, and part of building that antibody army that we want is the fact that we may have fever, we may have chills, we may have headache, we may have fatigue, but you're not sick with corona. And in every clinical trial out there, nothing really lasted beyond a 36-hour period, with the majority of people having symptoms resolved by a 24-hour period. And so when you get the second shot, we give you the first one and your body kind of gets a peek at it. It does begin building antibodies a little bit, but by the time we give you the second shot, we're now charging and telling the body, yeah, it's a real thing. We want you to build antibodies. And so now the second shot is always potentially going to be a little bit worse than the first one. And I can tell you, I didn't have any reaction the first time. And on the second one, I had a little bit more of a headache, a little bit more fatigue. But again, that lasted for about a day. And now I've gone back and tested. I have full antibodies right now. Now, the third one, about the third potential Moderna, that third potential Moderna shot, it was not because the first two did not work. What both Moderna and Pfizer are working on right now is they have the genetic code for the current variants that are on the market. And they are now building, and it only took them a 2-week period, by the way. Once they have the genetic code, they can tailor a vaccine to that genetic code of what they want the vaccine to work against. And that's really what they're working on right now. So it's not that the first two are not working. During that short video before it cut off, it talked about the spike head having 3 parts to it. The current thinking right now is that even that the mutations are occurring in one of the 3 portions of that spike protein, but current polyclonal antibodies are still attaching onto multiple parts of that spike protein. And so the currently available vaccines are still effective. Now, they may not be as effective, but nobody's really getting severely ill from any one of the, no matter what variant that they're having, if they've already had the vaccine. So I hope I kind of addressed that third vaccine thing. It's not that the first two didn't work. They're trying to tailor it again, just in case.
Okay. All right, Ms. Jackson, you want to go back to one of your questions?
Dr. Hill, I was just kind of combing through. I'm going to let you all bring the questions to me, but I saw a question here about the Tylenol.
Yeah.
Let me talk a little bit more about that. The Tylenol, we don't believe is a problem. What we do believe could be a little bit more problematic is Motrin and/or Aleve or aspirin. What we refer to as NSAIDs, non-steroidal anti-inflammatory drugs. So the Tylenol doesn't have this anti-inflammatory effect. So I'm not aware that people are not recommending Tylenol in all of the clinical trials that were done. Even if you look overseas, their version of acetaminophen or Tylenol is called paracetamol. They began using that in the clinical trials, and it did not appear to affect the antibody production whatsoever. But we are a little bit concerned about the anti-inflammatory effect of ibuprofen, of naproxen, or aspirin. So we're recommending people not take that. If you're going to take anything at all, just stick with Tylenol. Very often, Dr. Hill, you and I talk about it. People will interchange Tylenol and Motrin in terms of terminology, thinking that, well, it's all about the same thing, but they really do work differently in the body. Okay, Ms. Jackson.
One of the questions was, should a person who previously had COVID still get the vaccine?
Yeah, yes, you should. We're recommending that, and I'll give you I'll give you my best coherent answer for that. Very often, the immune response that you have, that an individual that had it, it can be due to a lot of different factors, okay? It could be how much viral load were you infected with initially? Did you have a very heavy dose or did you have a small dose? What was your nutritional status at the time that you became infected? I mean, there's a lot of different things that go into how you develop your particular antibody response, which will be different from one individual to another. Okay, and not everybody's going to have the very same type of immune response depending on the conditions under which they became infected. And we know that from having studied convalescent plasma. People that had a pretty heavy viral load, they tended to produce a lot more antibodies to give to convalescent plasma people, as opposed to people that did not have as much, and then clearly did not have nearly as robust a response with antibodies. Now, also having said that, so if you've been infected, we want you to go and get vaccinated again. The current recommendation is if you had it within a 90-day period, you can probably wait. But if you were infected more than 90 days ago, then yes, you can come back in and get revaccinated, and we encourage that. And now you're going to have a very known amount of vaccine coming in that should produce within normal limits about a similar response for every individual. Very good question.
Yeah. Thank you, Dr. Sneed. Well, moving along, one question is, should we get tested for the antibodies if we had one or more negative COVID tests, while others in the household may have tested positive?
An antibody test?
Yeah.
Yeah, you know, antibody testing is very, very tricky, to be perfectly honest. You know, antibody testing is probably more effective for people who were asymptomatic carriers. And if you were just curious and you think, well, you know, maybe I was an asymptomatic carrier, you may want to go and get antibody tested. Exposure to an individual doesn't simply mean that you're going to build an antibody response at all. And again, when you go and get an antibody test, it's only quantitative. It's not qualitative. We don't know if you really have a good quality amount of antibody, or a poor response to it. So maybe it's more qualitative than quantitative, but we're just detecting, do you have it or not? We're not detecting how many and how robust they are. And so that will be a very difficult question to answer, simply because, you know, the antibody testing right now doesn't mean a whole lot in terms of what may or may not have happened, what your future course may or may not be. Now, I can tell you, if you became ill with COVID and we knew you were ill with COVID and you tested positive for antibodies after that, that can be a very important indicator in terms of you know, your ability to contract it in the future. And we do antibody testing now for many of our, and not for all, but for many of our athletes here at USF. But the antibody test is still to be determined in my mind in terms of the true diagnostic value for what we're trying to accomplish.
There's a question. There's a one about the actual technology of the mRNA. It asks, does the mRNA in the vaccine code for S1 or S2, or for the antibodies against S1/S2?
That's a very good question. So the it's coding for the actual S1 that's on the very top, not the stalk portion of that spike. And that's what they actually attach onto. And remember, there were 3 different heads there on that S1 stalk. And so when I talked about a polyclonal response, that's kind of what we were kind of referring to. And the messenger RNA, the current one, was actually geared towards being able to address the all of the 3-headed spikes on the spike protein. And the mutations that are occurring now in the spike, in those 3 heads of the spike protein. And so that's why we're really concerned that, you know, we may get to a point where the antibodies are not as effective as we would want them to be. They may be attaching to portions of it, but it may not get the full closure of all of the spikes on the current virus in the way that we would anticipate. Now, I also want to share with all of you the mutations that are occurring. They're occurring because, you know, we're continuing to have transmission. And if you really pay attention to what's happening with the variants around the world, not just in our country, we had one variant that occurred, the B117, that occurred in the UK. We had the N501 that occurred in South Africa. We had a P version that occurred down in South America. And then we also have one that has now occurred, and I can't remember, C something in California. I really believe that we're going to find eventually, I don't think we have the diagnostic capability today, that regional transmission, according to the environment, really will play a big factor in terms of the ability of how it will mutate to adjust to the human beings living in that region. But the one thing I must tell all of you is that the more we transmit it, the more it will learn. It has learned. And we have to also keep in mind one more thing. the current COVID-19, the original COVID-19, we believe was a zoonotic transmission from animal that jumped over to human. But now the mutations that are occurring because of human to human transmission. So it appears to be learning us, and it appears to be learning how to adjust and adapt to all of us as human beings, making it more efficient in terms of how it enters a cell, and then having much more easily more transmission from that point. And so even after you've been vaccinated, we want people wearing a mask. We want to shut down all transmission if possible. And that's about the best I can tell you right now.
Okay. Dr. Sneed, it appears, that it's not clear to people exactly how it is that the vaccine is giving us protection, and we say that we're not giving a part of the actual COVID virus. So could you allude to that and explain that a little further?
Absolutely. Very good question. And the response I give you around the messenger RNA vaccine will be very similar for the Johnson & Johnson adeno-vector vaccine. And so basically what we did, or what they did, they took the genetic sequence of the virus, and they got the entire genetic sequence. I mean, what I call Ancestry.com or 23andMe, they got the entire sequence, and they were able to determine what part of that genetic sequence was specific to the spike protein. And then they took that portion and then they duplicated that in a laboratory. Okay, they were able to duplicate that in a laboratory, that part of the RNA, okay, strand, just that small part for the red spike that you find. Then they took that and they found a way to manufacture and reproduce it from one strand to the other. And then when we inject that messenger RNA into your body, okay, came from the laboratory, not from the original virus. We're not growing viruses. We're kind of manufacturing a small portion of the messenger RNA. When we inject that into your body will then recognize that, and it will use the messenger RNA to code and have your proteins in your cells manufacture that red spike. So you're literally growing a little portion, a really small amount of that red spike inside of your cell. You're not growing the entire virus, just the red spike. And you cannot get sick from the red spike. The red spike does not make you ill. What makes you ill from the actual virus is the RNA strand on the inside of it. Okay, that's the part that is really dangerous about it. But when your body recognizes that red spike, it builds antibodies against that spike. So if you become infected later and with the actual virus, now you have antibodies that are going to recognize that spike from the actual virus, and they're going to attack it, they're going to bind to it, they're going to neutralize it, and then eventually the virus will die because it will not have a chance to replicate inside of your body. And so we're not taking any portion of the actual virus right now. Many other vaccines, we do take an inactivated portion of the virus and we chop it up chemically and we inject you with that. Well, no portion of that is happening right now. Everything has been manufactured in the laboratory, and we're coding it, and we're kind of telling your body exactly how to make that protein so you can build antibodies against it.
Okay. Well, thank you, Dr. Sneed. All right, Ms. Jackson.
What is the window time to get immune after being vaccinated?
That's another very good question. Whoever asked that question, and you're going to hear and you're going to hear a lot of controversy. I'm just going to warn you. You're going to hear a lot of controversy coming out about this over the next couple of weeks here, and it's already started now. According to the clinical trial for both companies, after the first vaccine, you get about 50% protection, okay? And that's been the general thinking. Now they're going back and they're looking at that data again, and they're watching what we call these Kaplan-Meier curves. And according to the Kaplan-Meier curve, they're now readjusting thinking and saying, well, maybe you really get 80% protection after the first vaccine. For right now, I'm going to stick with telling people 50% protection after shot number 1. When you get shot number 2, clearly you get the full whatever you're capable of producing as an individual, about a 10 to 14 day period after that. Now, as soon as 7, but if we kind of round up, about a 10-day period after vaccine number 2 is when you get the full amount of protection that you're probably going to get from that 2-shot strategy with either Moderna or Pfizer. And so if you talk to me, I'm going to tell you, yeah, within a week, you're going to have full protection. If we really want to take a look at the data, probably a 10-day period. Other people may be a little bit more conservative and say, no, a 14-day period. But I'll go on a limb and go right in the middle and say at about a 10-day period, you should have about the full amount of protection you expect to get from that vaccine.
Okay, so Dr. Sneed, a lot of people are working from home.
Yep.
Should they wait until they're getting ready to go back into the work world to get vaccinated, or would you suggest going ahead and doing it now?
If you have the capability of getting vaccinated, you go get it right now, even if you're working from home. The actual fact of the matter is, until we shut down community transmission, nobody's going to be safe. And even after we vaccinate a number of people, just like with the flu or anything else, the virus will still be out in the community. So even if you're at home, you may leave to go to the grocery store, go to the gas station, or whatever it may be. No, go get vaccinated. If you have the capability, go get vaccinated. We have to do everything we can to shut down transmission. And by the way, and I've been sharing this with Dr. Hill, and this is from a health equity standpoint in terms of demographics. Okay, and it doesn't matter what demographic we're going to look at. It can be African American, it can be older patients 75 and above, it can be Latino, people of a certain socioeconomic region that they're living in. What's going to wind up happening eventually? When we get, I don't know what the actual number will be, but we're going to get a fairly large number of people who are vaccinated in our society. And at that point, people are going to start opening up, okay? More things are going to open up, and we're going to expect people to engage more. People are going to be more confident. Some other people are going to say, look, I'm already protected. I don't need to shelter anymore. I don't need to worry anymore. What will wind up happening if we don't get enough people vaccinated quickly enough is that the viral pressure will fall into the people who are unprotected. The virus will still look for people to infect. And so I'm telling people right now, if you have an opportunity to get vaccinated, even if you're working from home, go do it. Because waiting means that the virus is simply waiting for you to come out into a society where other people are going to feel much more comfortable about opening up and going back to restaurants and getting back to their job and doing much more than we're capable of doing today. Yeah, great question.
Well, we have several.
I saw a question here and from Len. Kaiser was recommending not to take Tylenol or antihistamines. Is that accurate? Again, I'm going to stand by what I've read, what I personally read in the clinical trials. Tylenol, acetaminophen did not appear to inhibit any antibody response. I did not read anything about antihistamines. I don't know how much antihistamines, whether it be Claritin or Zyrtec or Allegra, or maybe even Benadryl. I'm not aware of an actual mechanism that would actually allow those to not allow your body to produce antibodies. I can't think of a mechanism of action for that to occur. Maybe Kaiser, and I've got very good friends at Kaiser. Maybe I'll reach out to them and say, well, what data are you all looking at? But the data I've been looking at would indicate that acetaminophen should be safe to take. And I have not heard a widespread thing from the CDC or anyone else to not take Tylenol following being vaccinated.
So, Dr. Sneed, to that point, we have a few questions inquiring about whether or not Benadryl or Zyrtec prior to getting the vaccine would be helpful.
I don't think it would be helpful, to be perfectly honest. Prophylactically, then there's no mechanism for them to be helpful. To be perfectly honest, we have to keep in mind once you get vaccinated and your body has now recognized. And let me create a different visual for all of you very quickly. I want you all to think about everybody here. You're on a computer, and if you were in Word, you would type the word vaccine into a Word document, and then you would send it to a printer. Okay, the computer code going from your computer to the printer is very much like the messenger RNA. Okay, in this case, the messenger RNA is going to a printer in your body. And in your body, that is what we call a ribosome. On your desk, I have a HP printer right here. In my body, I have a ribosome printer, and it's telling the printer, in your case, to print a piece of paper that has the word vaccine on it. In the messenger RNA world, and even the adenovector, is coding and telling the ribosome in your cell to print that little spike protein. So I want to give a different visual metaphor for what is really happening. And so that the messenger RNA does not interact with your DNA. People are worried about that. They've wondered about that. It cannot change your DNA. It cannot alter DNA. Nothing like that will occur. And much like after you hit enter and you send it to a printer, that code goes away. It's not going to keep circling back and keep telling your printer, hopefully, to keep printing the word vaccine on a new piece of paper. The messenger RNA will degrade within hours. By the 12-hour mark, that messenger RNA is gone. It has told your body what to do. Your body is now manufacturing the little red spike proteins, and now your body will begin manufacturing the antibodies. And so I cannot think of anything around an antihistamine that would stop your body from producing antibodies. When we take an antihistamine, we're trying to shut down the histamine release that comes when we become infected with a virus, not the actual antibodies to the virus. Dr.
So
Hill, did I explain that okay?
I think you did pretty good.
Okay.
We're right at 4:30, and I wanted to ask Mr. Orlando was pleased. He put a thumb up.
Okay.
I wanted to ask the group if they wanted to continue. We have quite a number of questions still remaining, and I don't know, Dr. Sneed, about your availability.
I've got time. I'm looking.
Okay.
I've got a little bit of additional time before I have to kind of move on to something probably in about 20 minutes from now. But if people have questions and they feel like I'm providing value, I would love to answer them.
Well, we're gonna start kind of integrating them and see if you could. I'd like to throw 2 or 3 at you at the same time that are common.
Well, Dr. Hill and Miss Jackson, almost at the risk of kind of doing self-marketing, everything I'm talking to you about is just really from having done an enormous amount of reading. As I tell Dr. Hill all the time, I've read about 300 peer-reviewed articles on COVID-19, everything from the cellular-based all the way to behavioral and social, and impacts on in various communities. And so I'm going to give you the best evidence I can. It really it boggles me. It does boggle me a little bit when they're saying that Kaiser is recommending not to take Tylenol. I'm going to have to really explore that because I haven't read anything that would indicate that we should not take Tylenol. We are beginning to believe about ibuprofen and Motrin, but I haven't seen anything about that. So I'm going to explore that a little bit more. But I am talking to you based on evidence of what we currently know. And if we don't know it, I'm not going to say it to you. But yeah, let's keep going.
All right, very good. So one of the other questions that I had, and then I think I'll be almost done with my list, was that the Gsk vaccine that's supposedly coming out in the fall, will that one have FDA approval since it's going through the typical channels as other approved vaccines? There seemed to have been some concern from some of the other questions about the fact that, the emergency use authorization with Pfizer and Moderna bypassed certain steps, and that it showed up, they showed up very quickly, and we may not necessarily have all the wrinkles out. Some of the animal testing and different things weren't completed as we normally know them to be. So if you could allude to.
Let me explain the process to you, and really explain how and or why we were able to get to vaccines really as quickly as we did. Number one, every vaccine had a predetermined number of people. So, for example, I'm going to make up a number. If they enrolled 100 people in each company, they said, when we get to about 40 people who have become infected with COVID, regardless of whether or not they were in the active group or the placebo group, when we get to 40 people in every company, we're going to take a pause and we're going to look at the 40 people, and we're going to try to figure out who got infected. And so when you look at the 40 people with Moderna and Pfizer, at about a 95% infection rate, 95% of the people, so I guess that would be right at about 38 people, 38 people that became infected, they were in the placebo group. And the other 2 people that got infected had actually been given the vaccine. And so every company has a predetermined number of people that when they become infected, they don't know if they got the placebo or they don't know if they got the actual vaccine. They just know that we have hit our target for the number of infected people. Then we're going to go and find out who got infected. That point number one, okay, that's the first milestone. That's the efficacy milestone. The safety milestone came in every company. Before we can submit to the FDA, there must be a 60-day period, a 2-month period beyond the time of the last individual that received the vaccine. And so that window has been consistent for every single company. And they have done that because historically in vaccine trials, if you were going to have a real problem, everything tended to show up within a 2-month period. And so on the safety part of that milestone, every company, they cannot submit anything to the FDA until they actually have a 2-month period beyond the final individual that's going to be evaluated by the FDA. And that will hold true for every company. Now, the reason they are allowed to submit, they may have told the FDA, we have a plan and a goal to admit 45,000 people, but we got and hit our benchmark data on 43,500 people. So we're going to submit the early portion of that data to you right now. But then they also took into account the people that were in the phase 2 portion of a study. So if you take a look, very often they'll talk about a phase 2/3 study. And they combine all the people, and they got the safety data from the early people, and they got the efficacy data from the phase 3 people, and then they go and they submit. So they really did not have a shortcut in the way that people think. GSK will probably follow through and gosh, I'm blanking on the other. I can't remember the other company right now. Well, anyway, there's about 6 companies out there that are really doing vaccine studies. Oh, by the way, Merck, yeah, they stopped theirs. They'll continue through, and there will not be a need for them to go to an EUA by the time they complete their trial, because we've already had, by that point, we will have a lot of product on the market at that point. So it's really not happening. And the only reason, by the way, we were able to get 30,000 people in Moderna, 45,000 people in the Pfizer study, another 30,000 people right now in the Johnson & Johnson, another 30,000 people that will be in the Novavax study. I forget the total number of people in in the AstraZeneca, Oxford group. It's not, at that point, The only reason we were capable of doing that is because we were in a pandemic, and there were a bunch of people around everywhere. Typically, a vaccine study would not be able to move that quickly, not because of any safety concern, but they would not have been able to achieve getting enough people infected to evaluate. Okay, they would not have had enough people out there to actually evaluate whether or not the vaccine really worked or not. And so the speed of the trial really happened because we were in a pandemic and we had millions of people around the world. I mean, 25 million plus people have been infected. And that's the only reason it occurred. Right now, the Moderna company, they have a Zika vaccine. It apparently has been shown to be pretty effective, but there's not that many people out there with Zika right now. So they cannot move forward with testing that particular vaccine in enough people until they have people with Zika to test it on. And so I just wanted to kind of clear that up. That's whoever asked that question, that was a fantastic question to try and figure out why the EUA came about. How did it come about? Why did we go through? Did we take any shortcuts? No, we didn't take any shortcuts at all. We just really took advantage of the fact that we had 25 million people around the globe that were infected.
Okay, Ms. Jackson, you want to ask a question?
Yes, I'll just do a couple more so that way most everybody can get back to work because I know we're going over a little bit. But one question I had that came up was, can you explain herd immunity and can you explain the dangers of herd immunity?
Yeah, I can explain both. And one portion of herd immunity is dangerous, and the other portion is not. The excuse me, the portion of herd immunity that is dangerous appeared to be the strategy that one of the people in the previous administration task force, the COVID task force, appeared to be endorsing. Meaning, what herd immunity is, and we can take anything, is that you have enough people within a particular group that all become infected with something, whatever it is, and they all build antibodies against it, such that now you have protected enough people so that the few people who did not get infected will no longer be at risk of being infected from the people that they're hanging out with or their community of people or animals or whatever it may be. Okay, so you have enough people that have enough protection such that you can no longer spread it to the people who were not infected. Now, the strategy, the dangerous part of it is just letting the wild type, the original virus, run rampant and infect everybody. And, some people, they figure, well, some people will die, but the majority of people will live. And at that point, we will have enough people infected with enough antibodies that we don't have to worry about anyone else dying or anyone else becoming infected. That was a poor strategy for exactly the reason I talked about earlier. You don't want that RNA strand passing from one individual to another. It has already mutated. And by the way, we've already had multiple mutations here in the United States. We were identifying them last year. The first mutation really occurred back over in California and New York. So it's not new. That's a very poor strategy because in the meantime, people were dying. Now, if we vaccinate people, the vaccination, again, is not transmitting the actual RNA from that virus into your body. It's just helping your body build up antibodies. We're getting the second part of the response. So if we can get about 75, 80% of people in the country vaccinated, well, now we won't worry about transmitting it as much to the other 20% of people that may not be vaccinated. So it really is, herd immunity really means, can we get enough people with enough antibodies to shut down transmission of that bacteria or virus so that it will not spread to other people? But how you get there is very important to understand. You can't have that. And Dr. Hill, Ms. Jackson, if I can add on to that very quickly, I didn't have time to talk about in the very beginning, but people that have diabetes, people that have high blood pressure, people that fall into the overweight and obese category, people that have these chronic conditions, these very common chronic conditions, if they become infected by the virus, they can turn on inflammatory proteins that begin to do all the organ damage that I showed you earlier on in my presentation. And I believe that's why we're having such a hard time right now in our African American, and now in particular, our Latino community, especially out in the LA County area. Because if we have a higher prevalence of diabetes, if we have a higher prevalence of high blood pressure, if we have a higher prevalence of being overweight, all of these are predisposing conditions for a very poor outcome should you become infected. And so anyone that may have that condition, we're telling people, please take your medication, try the best you can to get your condition under control before you become infected. And by and large, they are the ones that wind up having a really hard time, that get placed on ventilators, and eventually probably wind up succumbing to death, much more than other people. Now, that's not everybody. Sunday evening, they had a story on 60 Minutes about a young man that he ran, I don't know, 5 miles every day of his life. He was in his early 40s, had no diabetes and no other condition, and he wound up dying. So there are other people that fall outside of that category. But people that fall in that category are particularly susceptible to having a poor outcome.
And my last question is about pregnant women and their ability to get the vaccine, or should they even get it?
The current recommendation is that if you are pregnant or if you are breastfeeding, that you not get the vaccine right now. Now, that's a recommendation in terms of electing to get the vaccine. I can share with you, and this is all public information, you can go and find it yourself. During the clinical trials, especially for Pfizer, a number of women became pregnant during the trial. And they have monitored all of those women during that entire time. And so far, they have not found any harm to the pregnant women or to the developing baby. Now, I have not heard or read a report about what happens after the babies have been born. But to be honest, with messenger RNA, we don't have a reason to believe that it would actually cause a problem. And I know that there's a lot of things on social media, on Facebook. There are a lot of people out there that are adamant that it's a problem. And as of right now, we don't have any evidence of it. And people that were pregnant, they became pregnant during the trial, and they are following and monitoring these people very, very, very closely. In the trial that we're doing right now, we don't allow pregnant women to come in. But I can promise you, if a woman becomes pregnant during the study, we're going to be bringing them in for a lot more visits and monitoring everything about them. And all that is going to be free care for that pregnant woman. And we're going to monitor everything we can. And we've already talked about it. We know the protocol. We know exactly how that will go. We don't want them to become pregnant, and we will not admit them into the trial if they are pregnant. But once they become pregnant, we have a duty and a responsibility to try and take care of them and to learn everything we can. And in the Pfizer study, that clearly did occur. But if you're pregnant, the recommendation right now is to not get vaccinated. If you're breastfeeding, the current recommendation is not to do it, but not because they found any harm in any of the trials. That's really the point.
Well, Dr.
Okay,
Stevens, I was going to say we do have 1 or 2 more questions, but I did put my email in the chat, and we had requests for copies of your slides, and I just wanted to let everyone know, in lieu of giving copies of the slides, because the information is so dynamic, we don't mind sharing the recording of today's workshop with you. And we also encourage you to attend some of our future workshops. This Thursday, we'll be revisiting the Tuskegee study and the Henrietta Lacks story, and talking a little bit more about the vaccine and the need for research consideration. And then we'll be having some more workshops later this month. So we'll share them with Ms. Jackson and others. And now that you all have registered for this workshop, we'll have your information as well. But we would like to thank all of you for attending today. Dr. Sneed, I think I can go on record to saying this was probably one of the highest attended workshops that we've done in a while. So we'd like to thank you, Ms. Jackson, for being so passionate about having us come to your group. It has been a pleasure, and we are certainly intrigued by the work that you all do, as well as impressed. And I will be continuing conversations with Ms. Jackson about how perhaps we can work together in the future. So thank all of you for all of your questions. And if you have any more, please feel free to reach out to us. And thank you, thank you, thank you. But stay safe, be well. And one of the things that Dr. Sneed did not really get a chance to say much, but I want to say, make sure you're wearing your masks, washing your hands, and socially distancing appropriately, and stay safe, stay well. And hopefully right now, although the priorities for the vaccine seem to be for individuals over 65 and for those that are essential workers or in the trenches as healthcare workers, the time will come, and we hope that when it does come, you'll consider getting vaccinated. So
yeah, just one more thing. I want to, once again, I want to thank Neo-Genomics for inviting me to come. I really hope I can find a way to work with you all, just so I can go around and tell people I'm working with Neo-Genomics. I mean, that's like the coolest name ever. But no, thank you all very much. I hope, if nothing else, I hope I've given many of you an opportunity to hear information that you may not have heard. You can go and explore anything I talked about on your own. Get it from, but if you get the information, get it from a credible source, and that's all we're asking. Try to get the information from a credible source, and stay safe. And Ms. Jackson, thank you so much for the opportunity.
Yes, and thank you guys so much for coming out and speaking to us. When I first heard this, I was so impressed that I had to share it with the rest of the group. So I'm so glad you guys took the time out to come and speak to us and get the information out.
Thank you.
You're welcome. Awesome.
Yes. Okay. Well, we'll be in touch. Definitely.
Okay.
Thank you. Take care.
Same to you. Bye.